Hello, welcome to CytoCave. It starts off with the default coordinate system isomap with the node degree color coding. There's two views, they're each independently configurable. You can select different coordinate systems. We have isomap random trees, t-SNE. The sphere represents all the proteins. This cloud of cubes represents all the drugs. Adjust the threshold down so we can see all the edges. If we search for an interesting drug like nilotinib, a cancer drug, you can see that it lights that one up, and it also lights up all the proteins on the left that, well, it lights up the edges from the central cube. It'll make these protein spheres a little smaller, so I'll unlock these locks so we can control the left and right sphere and cube sizes independently. So the central cube in the center represents all just drugs in general, so all the drugs. So since we selected this drug here, nilotinib, you can see it projects rays to all of the proteins that it affects in this red and green cluster of spheres on the right here, in the t-SNE coordinate system, but on the left in the isomap coordinate system, the drugs are all collapsed into the central cube in the origin, so you can see how that drug, or if we select other drugs, how those drugs represent how they affect all the different proteins. Let's make that cube a little smaller and make the protein spheres a little bigger. When you hover over a protein or any node, there's a label that can come up that points it out. This is useful in VR, but it's still experimental. So it gets further away and it gets obscured by other nodes. So there's CDK4. This is a cyclin-dependent kinase. So that this drug, nilotinib, is shown here to affect CDK4. There's PARP1. Apparently the drug does not affect PARP1, but it does affect CDK4. Now if we select bosutinib, now we have two drugs selected on the drug side over here. There's bosutinib, there's nilotinib, and you can see they're projecting their rays to all of their proteins. So it's also useful to have the same coordinate system on both sides. So this way you can see, you can sync them up. You can unlock the color coding and you can have them represented in different color codings. So we have degree range on the left, and we can do the native t-SNE on the right, so we can see because the we use DB scan to cluster to assign module membership to cluster to nodes based on their positions, so that in the coordinate system, and try to identify clusters. So if you have the t-SNE coordinate system and the t-SNE color coding, then the clusters should show up because they're coherent. You can also select Louvain cluster color coding, cluster color coding, and this finds clusters independently of the coordinate system. So you can kind of see how those overlap. So if you have the native t-SNE on one side and the, say, Louvain on the left, and then t-SNE on the right, you can see how the drugs, the drug networks, the clusterings overlap. You can also zoom in on the genes. You can do that on both sides. You can see the t-SNE and Louvain color codings. Make these boxes a little smaller so you can see the edges. So there's a central blue cluster with a red periphery for the t-SNE and DB scan coordinate system and clustering algorithm. But for the Louvain algorithm, there's two central clusters and different kinds of clusters on the periphery. Now, you can control the threshold of edges because the edges correspond to interaction strengths. So if you select this slider and slide it over to the right, you're increasing the threshold of interaction level that determines an edge. So you can slide that and you can see these, how they appear and disappear. You can also click the intra or intergroup checkboxes. So right now we're seeing both inter and intragroup edges. If you want to focus on intragroup edges, so you can uncheck intergroup and you can see just the protein interaction networks, or there's also the drug interaction network. Select some more drugs to show those. Or if you want to just focus on intergroup connections, you can turn off the intragroup ones and you can see pretty much the drug and protein interaction, so you can really see which proteins are affected by which drugs without having the clutter of seeing all the protein-protein interactions. And turn all the edges off. Turn all back on. You can adjust the opacity of the edges so you can they're a little more translucent. You can, maybe they don't obscure one another. You can see them better and you can see which nodes they point out better. And you can also control which modules are visible. So you can toggle, like say a module number 2 can be toggled, make it translucent, transparent, or visible again. And that shows up in both views. You can also toggle all of the edges, all of the nodes, and then so you can turn them all translucent. And if we're interested in module number 2 more, you can make that stand out by making it fully opaque against a translucent background of all the other nodes. And I also wanted to show you these sunflower representations. So this is the random trees sunflower. It's a dynamically generated layout generated from the module membership. So you can see random trees there, and maybe the Louvain modules. This shows you it lays out the nodes in sunflowers on the faces of a platonic solid with the appropriate number of faces for the number of clusters that there are. So it brings all the clusters together and shows how the clusters affect one another. And the clusters can be turned on and off through here too. You can adjust the opacity of the edges. And then adjust the color coding. So it's Louvain. So the colors will actually match the modules.